Okay, so I wanted to say a bit about reporting standards and why they are useful and what we have been suggesting to use. So it's a bit of a different talk from the others earlier today, where you learned a lot about methods. This is more about reporting. But I think we have a big problem with working in environments that the observations we are making are just points in a large area and at very few time points as well. And the environment is complex and changes all the time. We have spatial variation. Variation over time law showed some time-dependent concentrations in London just in the last talk. And there are lots of environmental conditions that will affect what we are observing, that we should also record these factors that could have an influence on our measures of interest. We have a huge diversity of microorganisms, of course. That means if you're taking a small sample, we will be missing rare members of the community with some probability. We will almost always capture the ones that are more abundant, but we will miss some rare bacteria, potentially ones with resistance genes. There are lots of different resistant genes, thousands of them, and other genes that aren't resistant genes but provide resistance as well. And there's tolerance and persistence and other things as well. And it all evolves. So we have also a complex mixture of potentially selective chemicals, not only antibiotics, but also disinfectants and metals and other things that could be selective. So it's very complex system to study. And we, of course, can't sample everywhere and all the time and measure everything. So we are ending up with pieces. And the difficulty is to bring these piecemeal observations together to come to some conclusions that are reliable. So we want to, have reliable And the pooled effect size, the odds ratio was 2.33 for the positive relationship between consumption and resistance. So more consumption means more resistance. And the odds ratio of 1 would be no effect. The odds ratio above 1 would be a positive effect, and this one is sufficiently above 1 to be statistically significant. And showing that across many different studies, there is a clear relationship between consumption of antibiotics and the level of resistance. This forest plot is I'm only showing about half of that figure from the paper because it wouldn't fit on the slide and still be readable. As you can see, there's a lot of variation. The solid line in the middle is the odds ratio of 1, which is no effect. The dashed line is the level where it becomes statistically significant, that the odds ratio is higher than 1. And you can see different studies. Some have huge variation, like these here. Some have reside with a negative relationship. Most of them are on the positive side. Some studies have a very low variation, and for that reason, in this pooled analysis, a high weight. This one here has the highest weight, I think. And they've actually taken it out. Still doesn't actually change the odds ratio too much if you take that study out with the highest weight. There are some other studies with low variation and therefore higher weight in this analysis. And overall, the odds ratio is 2.3 and statistically significant. So this is what you can do in a meta-analysis if you find enough paper that use a similar enough methodology to be pooled. Okay, now our colleagues in Israel, Nasrin Hassoun-Kir, Yoav Starpolt, and Michal Paul have wanted to do a meta-analysis of all studies comparing hospital and community wastewaters in terms of AMR. We helped a little bit in this, but mainly the work was done by Nasrin and Yoav as reviewing the paper, and Michal as the moderator if needed. They are all clinical microbiologists, infectious disease experts, so they have done a lot of systematic reviews in the past on more hospital-related studies, and were quite shocked by the heterogeneity of results and methods in environmental studies. So some of the main findings were that the reporting on sampling methods and wastewater treatment processes and statistical analysis were at high risk of bias. So there was very little information and different information available in different papers. So the reporting was really too poor to be useful for a meta-analysis, and the heterogeneity methods also made it impossible to do a meta-analysis. What we wanted to do, we couldn't do, so we ended up doing only a systematic review. That came out last year. And the frustration from this was that really reporting in these papers was not allowing us, and the differences in what they reported didn't allow us to do meta-analysis. So as a consequence, we decided, I wanted to show a few more results from this review to explain it a little bit better what I mean by heterogeneity methods. So this is one example about vancomycin-resistant enterococci in wastewater, and just looking at the different ways in which the results were reported, you can see that you cannot really combine the data. One way of reporting the results was taking the log counts per volume, so an actual absolute abundance of the vancomycin-resistant enterococci. Another common one was the number of positive samples out of all samples. Now that's a very different way of reporting this, and depends a lot on the volume of the sample. You know, the larger your volume, the more likely you're going to detect something that's present only is very rare in the sample. And positive or negative is just about detecting whether there's at least one in there or not. So depending on sample volume, of course, sometimes divided by the number of sampling locations when there was no information given on the number of sampling locations. Not a very useful way of reporting things. Or percent VRE, not very clear what that means. So you see 4 or 5 different ways of reporting what the data, which makes it impossible to use in any meta-analysis. Another example, again, I've cut out about half of the table because there wasn't enough space on the slide, otherwise you couldn't read anything. So this is the risk of bias analysis here for different categories, sampling comparability and analysis. For many different studies, you can see that if you look down columns, you can see that outcome units are quite often a problem. Statistical analysis quite often a problem. If you look sideways, there are some studies that have more green and others more red. So there's lots of variation. U means unclear, and blank means not applicable. So lots of variation between different studies. So as a consequence of this, we decided to come up with reporting standards. And in order to draw up reporting standards, we gathered input from a number of different researchers. In total, we had at the end 21 experts giving input into what was new to me, the multi-step modified Delphi consensus procedure, which is basically a mechanism to ensure that there is agreement between all experts on each particular item. It wasn't too difficult, but anyway, the procedure might have hopefully also worked with if there had been more differences of opinion. We are now calling this reporting standard Embrace Waters. It's an acronym that is difficult to remember for what it actually stands for, reporting antimicrobial resistance in waters. So Embrace Waters, that has come out a few weeks ago in One Health. Lots of authors, because all 21 experts are on that paper. Thanks all of them for contributing. And I just wanted to spend some time going through our recommendations on reporting. Some of this will be obvious, some will be less obvious. So I spend more time, I guess, on those which are less obvious. This is, there are 21 in total, so I put it on 2 slides. Title, abstract, and background. This is kind of the normal things, what you would expect. Describe the environmental compartment and resistance study. So already in the title, you should be clear about what system have you studied and what exactly have you looked for. A recommendation of a structured summary. And the background we described, well, the background and the rationale of the study, that's not surprising to anyone who's written the paper. Now, in terms of the methods, so this is about planned locations, so locations that were planned before starting the study. or before taking the samples. You should report on predefined sampling location, give a rationale for why you've chosen particular locations. And then describe the sample types in each location. Was it a water sample? Was it a sediment sample? Was it some other kind of sample? Be very clear about that. And the technique employed to sample, like, you know, a Laura's bucket grab sampling, or the passive sampling that she described, totally different results, or continuous composite samplers, you know, different methods of sampling will give you different results. So it's got to be very clearly explained how you took the samples, what equipment you use for sampling, and the volumes you sampled, because that is important to report the volumes, because a large volume is more likely to capture the heterogeneity of the environmental system. And then the sample processing for each sample type, and whether you took any steps on site to preserve the sample, like cooling or not, and how long did it take to transport to the lab, and all these things that can have an effect on the results need to be clearly reported. Source characterization, that's the point we have probably most discussions on, especially with the reviewers. Two different cases here. If there's a point source, there's no point source. So if there's a point source, such as a wastewater treatment plant effluent, or another inlet with some material, or from a farm, if you have a point source, you need to describe the point source as much as you can and the characteristics of the point source. And if you don't have a point source, you should describe the characteristics of the watershed from which the runoff enters the river, for example. So this is important, and I understand not always easy, but it is worth the effort of trying to find out exactly what is this wastewater treatment plant doing, what capacity does it have, etc. What treatment process does it use, or what farmland, what livestock was potentially contaminating the water. Then coming to the microbiological methods, describe the methods used to detect bacteria, how resistance was assessed, was it phenotypic, was it sequencing, or both, and the statistical analysis and analysis pipeline, what you plan to do, and sample number calculation is recommended as well. Now, coming to the results, you should report the actual locations sampled with GPS coordinates, so be precise and give the precise location, but also the kind of location. What is this? Is this a bridge, or is it the riverbank? All these things that influence the way you sample and what you sample. Reporting the season and dates and frequency of sampling. Surprisingly, it's not always reported the dates or the season, or any kind of idea of what the conditions were like at the time of sampling. And importantly, if you're taking a sample from the river, it doesn't matter so much what the weather is right at the time to take the sample, but what the weather was like in the previous days leading up to the sample. Because what you take out of your river has gotten into the river before, and whether there was a rain yesterday that washed stuff into the river or not will have an effect. So you need to describe the weather conditions leading up to the sampling, depending on how long back the effect might be. You should generally measure and report water quality parameters and any other metadata, and report all these results per location. Any very important to avoid bias, include any negative results. Report everything sampled and measured. The units of analysis should ideally be concentration units and not absence, presence, or something like that, and have confidence intervals or error bars of some sort. Describe statistical analysis again. That is not surprising, I hope, to anyone. And discussion again, discuss findings in context of existing evidence, compare how what you found is consistent or inconsistent with other studies, and importantly, be clear about the limitations of your study. Okay, so that's 21 recommendations with some sub-points here, so a little bit more maybe than 21. And I wanted to show some examples, more or less randomly, not systematically reviewed, more randomly picked from the literature. Okay, I think I'll blend in the next one. Okay, so I guess you can see that within about the same number of words, there's a lot more detailed information on the right-hand example, despite the fact that it's a more complicated thing to coordinate 10 different countries and 16 different treatment plants. It seems like very well organized sampling campaign and clearly described. Whereas here there aren't many samples sometimes. There is quite some description about the rivers and why these are sampled and so on, but it is quite unclear and a bit unsystematic the way this was sampled. What I also liked is that they were actually clear about taking the samples 10 times downstream from the effluent, 10 times the width of the river. Now, actually, this is something that Laura already mentioned, the fact that rivers, if the wider they are, the less mixed they may be, and the longer it takes for them to become mixed. And actually, one advantage of being now in a team of people including a number of hydrologists, that I've actually learned something very useful from my hydrologists. There's a rule of thumb, but this is only a rule of thumb for the mixing length. It depends on the width of the river. So the wider the river, the longer it takes for an influent point source to be mixed into the river. It also depends on the depth. So the deeper river makes it easier to mix laterally. So a wide, shallow river mixes least, and a more narrow, deeper river mixes more rapidly over a shorter distance. So you can use this to decide how far down from a point source should you take a sample, where you can expect the sample to be representative, or the grab sample to be representative of what's in the river. So very useful information I learned from Josh Larson from my team. Two more examples of planning sampling sites that I liked, because they were quite clear and clearly explained. One example here, showing one wastewater treatment plant and upstream and downstream sampling sites dotted along here at quite frequent, quite closely spaced. Also including flow measurements, so having some idea of how much discharge from wastewater treatment plant compared to the river. And you can see in this case, the river is quite small. So no more than 5 times the volume of water, the discharge then from the treatment plant, and sometimes only twice. So the dilution of the effluent will be very small. A point the other paper is also making here with the dilution factors, this is very nice, but means that you also have to know the discharge. So have some measure of flow to be able to work out these dilution factors, but I do think this is really useful. And here you can see sampling down the river where they have different sampling points after the point source. And things change, of course. There is quite a rapid change for the first few kilometers, and then not too much happens after that. Once you know this, you can plan your sampling better to get the right kind of distances from the point source to observe where things actually change. I think both of these examples are quite good ways of planning the sampling locations and reporting them, especially the fact that they used flow measurements. And for that reason, we're able to work out the dilution of any point source. So to finish, I wanted to plead with everyone, including my own team, that we should be agreeing on these reporting standards for our own work. There may well be a need for modifying these standards or improving them, and I'm sure we will all be happy to improve these reporting standards based on any input or comments you may have. The idea is to have a kind of web page for updates of this. We haven't done that yet, but we should really be doing that. So if you do have any comments or suggestions for improvements, do get in touch. It would be great if you could agree on a core set of things to measure in all projects that we can compare across projects, like, for example, measuring absolute concentrations of E. coli as a sentinel organisms or an E. coli specific gene, to quantify bacteria, quantify genes, quantify chemicals, and water quality parameters sort of for environmental conditions. What's the oxygen concentration? What's the pH, salinity, all of these things. Can we agree on a common methodology? That's the question mark here. I hope that we can agree on some core common methodology and allow some variation, of course, for things that are project specific, where there will be different methods used or different methods tried or developed, of course, but that we have a common core of methods that we can use for for combining results across projects, and where we do use different methods, that we are aiming to cross-validate them. Yeah, I think I want to have enough time for discussion of these points. So one of our program coordination team tasks is in fact the cross-validation. As one of my tasks, including organizing these workshops, which I more and more realize have been extremely useful so far, and will continue doing those. Ideally, we would do something like the paper that Laura mentioned from Diane Agar's group on harmonizing methods across different groups. I think that would be useful to do if we can, but at least have some cross-validation of our methods. Yeah, the paper that Laura mentioned comes from this consortium called HERD. Yeah, I think that was all.